Okay, good evening everyone. So let me share my slide. Then we can start. Okay, yeah. Now I think my slide is visible. Let me take the pen. So okay, let's start. So good evening everyone. Myself Swankar from Indian Institute of Science, Bangalore. So I am one of the TA of this course. So this is the NPTEL online course on analytical chemistry. The course instructor of this course is Professor Devashish Ray from IIT Kharagpur. So this is the last class of this course. So whatever we have discussed from earlier class, that what is the application of this analytical chemistry along with this different type of thermal method as well as spectroscopic method, how we can use this spectroscopic method. Suppose you get some sample, unknown sample, and you can analyze, or you can get what is the composition and what the element is there, and you can get the information about the element and what is the concentration of that element in that sample. So all these things we can what is called get step by step, and along with this we have discussed about the electrochemical method and how we can use this electrochemical method to analyze the sample, different type of material chemistry as well as biological chemistry or bioelectrochemistry. So all these things we have discussed little bit. So today we will discuss about the application part of this analytical chemistry as well as electrochemistry part. In this course, basically 5 video it is available in this course, so if people can watch. So basically last class what we have discussed about the cyclic voltammetry. So basically cyclic voltammetry, if you see that these things, the current versus potential, you will see this type of what is called graph is we can get or observe. So where we have discussed already that why this type of graph or duck shape graph is there. So basically we are starting from here and it is going to the high potential. This is the US convention where the upper part is due to the reduction and lower the oxidation. So basically we have discussed two type of convention. One is IUPAC convention where the potential is lower, potential is the left side and higher potential on the right side, and this is the US convention where you can see the lower potential is the right side and the higher potential on the left side. So here we have observed that the C point where the maximum reduction is going on or reduction process is going on, and this is, as we have discussed, there is three type of current is there. One is convection and other one is migration, so and other one is diffusion. So these three processes there, basically cyclic voltammogram, which we have already discussed that this is a diffusion control process where you can see this is the maximum diffusion process, and after that the double layer or is forming. So the diffusion layer of the analyte is increasing. So after that the pieces from the bulk solution to your electrode surface, the probability is decreasing. For this, after maximum current it is depleting, and after it is going to the D point, and after that it is shifted a little bit because the FeFC whatever is there, it is in the bulk solution, so it is coming again to the electrosurface. So it is, there is some time gap is there. For this it is there, it is not exactly on the opposite or opposition or the same position of the potential whatever the reduction is going on. And this B and E point, which is known as E half, which is also known as the formal potential, and the formal potential basically the potential of the EPC and cathodic process and EPA anodic process by 2. So this is the key formal potential. So all these things we have discussed, and this is basically the electrochemical cell where basically you have the working electrode. This is kindly glassy carbon, and this is the counter electrode, and this is the reference electrode. So we discussed that when you are measuring the potential with respect to the reference electrode, where the counter electrode, the function is that you are transferring the electron through the counter electrode. As a result, you are measuring the current respect to the counter electrode, and you are measuring the potential respect to the reference electrode. So whatever potential you are getting, that is a exact potential. Okay, so these things we have discussed. Then we have discussed that what type of solvent we should take, or what is the characteristic features should be there for the solvent. So because we have dissolved the electroactive pieces on the solvent, so what is the characteristic of the solvent? So that it should be the temperature, whatever temperature you are doing the experiment, that should be stable in that temperature, and it should be inactive in that redox potential window, whatever potential window. Suppose you are doing the redox reaction on that this potential, so your solvent should be stable in this potential. And other thing is the what is called oxidase towards the stable towards the oxidized and reduced species, whatever reduction or oxidation product is forming, that should be stable against that potential against that product, and it does not lead to the any reaction with the pieces, whatever pieces is going what is called forming, and it should be inert towards the supporting electrolyte. So now question, what we can take the electrolyte or supporting chemical. So supporting chemical should be also soluble in the solvent, and it should be chemically and electrochemically inert, and it must be purified. So after that we have discussed that what should be the step to acquire data. So that is the thing. So first we have to do the background of the solvent and the supporting electrolyte. So then we can see that okay, whatever potential we are doing the measurement for your analyte, in that potential window your supporting electrolyte as well as your solvent should be inert condition, and we have to do suppose you are doing some reaction, we should do in the inert atmosphere, because in the there is some dissolved oxygen or some other atmospheric gas like carbon dioxide or CO is there, which can interfere in your reaction. So there are things we should take care. For this we have to do the any electrochemical reaction in inert condition. And other things is measure the open circuit potential. Basically we are starting for the A point, so we don't know where should we start first the what is called scanning key process. So for this usually whatever we are doing the electrochemical process, we should start from the ground state. So for this we should measure the open circuit potential, which is the potential of the between the working electrode and reference electrode when you are not applying any potential, external potential basically. So these things we have discussed, and other things is the ohmic drop. So there is basically the resistance between working electrode and the counter electrode, which is known as the solution resistance, and in this there is two part. One is uncompensated resistance and another one is compensated resistance, which can take care by the instrument. But this uncompensated resistance basically between the resistance between the working electrode and the reference electrode. So this due to this resistance, whatever potential we are getting, that there is some depletion is there from the exact potential. For this we should do some impedance spectra to know the resistance between the working electrode and reference electrode. So from there you can calculate the IR drop or ohmic drop. So from there you can get the information. Okay, so these things we have discussed. Now the today class we will discuss about one specific electrode, which is called Clark oxygen electrode. So basically Clark oxygen electrode, whatever we know that in Clark oxygen electrode there is two electrodes there. One is platinum, which is a cathode, and this silver, that is the anode. So in these two electrodes are dipped in the KCl solution. So basically to know this Clark oxygen, to know the dissolved oxygen in your solution or whatever blood sample, suppose you how much oxygen is dissolved in the blood sample or any biological liquid. So to know that information, this is one particular type of innovation or specific electrode, which is known as the Clark oxygen electrode. So if you see here, this one, if you zoom out, then you see there is some Teflon membrane is there, which can only pass the oxygen from the solution to the particular electrode, and the here oxygen can reduce to water molecule, which is a 4 electron process. So this platinum electrode surface, this oxygen can reduce to the 4 electron process, through the 4 electron process, and water molecule can form. So from there you can get how much oxygen is dissolved in this case solution. So these things is there. Let me check one thing. Yeah. So and other thing is these things is there. So how you can measure this oxygen dissolved? So basically here silver where whatever is there, this is the anode, which I have already told, and this platinum as a cathode, and this so oxygen is dissolved in the electrolyte. So basically we are dipping this portion in the whatever support sample. Suppose you have blood sample, and inside in the blood sample we can dissolve or dip in the electrolyte, this electrode in the blood sample, and you can measure the current in this potential voltage known as the magnitude of 700 millivolt to reduce this oxygen to water molecule. So and whatever current you can observe, these things you can measure. So basically whatever voltammogram we have measured, that is we are measuring scanning the potential, and potential scanning this potential, and we are measuring the current. So here also one kind of determine the diffusion oxygen rather than the voltage, you can measure the whatever current that is proportional to the oxygen concentration. Okay, so the Plato correlated to the oxygen tension in the electrolyte. So these things is there, and then question is what is happening on the cathode and what is happening on the anode. So basically cathode anode is the silver, and so whatever silver is there, and it is deep in the saturated KCl solution. So silver is oxidizing and Ag plus is forming, so it is react with the Cl minus and it is from AgCl. So this is 4 electron process, and this 4 electron can transfer through the outer circuit, and it is going to the in your cathode, and in the cathode what is happening, your oxygen is diffused through the Teflon tape, and it is going to the electrode surface. So this oxygen can reduce to from 4 electron process to water molecule. So what is the overall reaction? The overall reaction is silver and this oxygen, and it is forming 4 proton, and 4 chlorine is forming 4 AgCl and 2 water molecule. So this is the key process. Now question, it is, although it is a great innovation, there is some limitation of this electrode. So what is the electrode limitation? So this electrode limitation is we are dipping this part in the electrolyte solution. So as this is so although there is some limitation is there. So what is the limitation? Limitation basically the foremost limitation is you are dipping the electrolyte. So this oxygen diffusion, which is not a control process, so it is going to the electrode surface. So and it is not a control diffusion process. As a result, the what is called restriction, unrestricted reduction of the oxygen is going on. As a result, whatever value we are getting from this electrode process, that is not a actual value. So this is what is called unrestricted, we can say the unrestricted oxygen diffusion. Oxygen diffusion. Other things is that we are putting on the what is called biological sample. So in the biological sample, some other protein molecule and other enzyme also there. So which can interfere in this reaction. So in the electrode surface also, this type of protein or other other enzyme molecule can oxidize or reduce, so it can interfere the oxygen reduction. So whatever data you'll get, that may not be actual things, but this is one of the greatest or great innovation to study the what is called level of oxygen level in the blood sample. So these things we know, and after that we have discussed about the RD and RRD. So RD basically rotating ring electrode, and RRD is the rotating ring disc electrode. So RD rotating, this is rotating disc electrode. So I think ring is, so this is disc, rotating disc electrode, and this is the rotating ring disc electrode. So basically rotating disc electrode is black one, that is the disc, and in the outer part the platinum, this is the platinum, and this is the glassy carbon. So glassy carbon disc, and this is platinum ring. So this is the RRD electrode, and this RRD electrode is used to measure the kinetics parameter of some redox reaction. So and this is the RRD instrument, where you can see that this electrode is connected here, and here you have a solution or of your analyte, and this is the counter electrode. So counter electrode, and this is your reference reference electrode. So reference electrode you can use if it is aqueous solution, then saturated caramel electrode or AgCl you can use. So this is, and this is the working electrode. So in the this part is the disc. So your you can connect this part to the instrument disc, and this side you have the another connection for the ring. Okay, so these things, this is the RRD electrode. So if you do some oxygen reduction reaction or oxygen evolution reaction, so basically ORR and OER, basically this type of LSP, you can see this type of reaction will going on. So basically this RRD electrode is a hydrodynamic process. so hydrodynamic process. Okay, so basically you are rotating your electrode. As a result, whatever electroactive pieces is there, that is coming from the bulk solution to the your electrode surface. So basically ORR and OER people usually use zero point 0.1 molar KOH solution, and here they have dissolved oxygen, purging the oxygen, so that oxygen going to the coming from the bulk solution to the electrode surface, and it can reduce two electron process to form peroxide, or it can another two electron process to form if it is a 4 electron process, then it can form water molecule. So this 4 electron process can be also be processed through what I can say step by process, that is 2 electron process. So that way also you can form, it can form. So whatever, if it is a 2 electron process, then you can fix the ring at a particular potential, so that water can reduce 2 electron process and it is form peroxide, and then if you fix the potential where the peroxide can oxidize, so that it can again form the oxygen, so oxygen evolution can perform at the ring electrode. So basically on disk electrode where your reduction process is going on, and ring electrode where your oxidation process is going on. Okay, so if you do some LSB, linear shift voltammetry, which is a one segment of the cyclic voltammetry, so you will see this type of graph. So where you can see there is three part is there. One is where very low potential, low current is there, which is known as the kinetically controlled region, where any electron process is not going on, and where the depletion is going on or current value is increasing. So this part is known as the mixed region, basically the kinetic region as well as the diffusion control region. And the far potential, which is lower potential, which is control diffusion control process. Basically your electroactive pieces is coming from the bulk solution to your electrode surface. So this is the what is called diffusion control region. So basically from here you can get different type of information like kinetic parameter, so which I will discuss in the next slide. So basically if you do in different rotation speed, then you will get this type of graph. So here you can see with what is called speed or with rotation, the current value is increasing. As the rotation increase, your electroactive pieces from the bulk solution to the electrode surface is increasing. So as you know that the current value is proportional to the concentration of the active material. So as the electroactive pieces is increasing on the electrode surface, your current value is increasing. So with rotation, as it is hydrodynamic process from the bulk solution to your electrode surface, your electroactive pieces is coming. So as a result, you can see that your current value with rotation is increasing. And other thing is the onset potential. We usually know in the onset potential, people tell about the onset potential. So what is the onset potential? Onset potential basically particular potential where your current value is increasing. So basically where the ferritic reaction is started, so that process is known as the onset potential. Okay, this thing is there. And from the diffusion control region, if you plot as a J, so one as it is a electrochemical process, so you can use Kotek-Levin's equation, which is one of the famous equation for study the hydrodynamic process. So basically the reciprocal of the current value, which is equal or summation of the reciprocal of the diffusion control current density and the kinetic control current density. So this kinetic control process can the diffusion process which is depend on this 1 by b omega. Omega is the rotation rotation speed, so which is in radian per second. So unit is radian per second. Here it is rpm, so the rotation per minute, and here radian per second. So this b value which is a constant, basically 0.62, and f d to the 2 by 3 d to 1 by minus 6, and c0 where n is the number of electron in the of this reaction, f is the Faraday constant, and d is the diffusion coefficient of that electroactive species, and nu is the kinetic viscosity of the electrolyte, and c is the bulk concentration of your electrolyte. So electroactive species. Okay, so basically if you know the diffusion coefficient and kinetic viscosity of your electroactive species, then from here you can get the number of electron what is in this process. So basically whatever I have shown in the previous slide here, so here basically I am telling that this is a 2 electron process or 4 electron process. So this oxygen reduction reaction, that is 4 electron process or 2 electron process, that you can get or you can calculate from this equation. So and for that we have to plot 1 by j. So basically 1 by j versus omega to the power square root of the omega. So from here you can get the slope, and from the slope you can get the number of electron, and from the intercept you can get the kinetic current density. So this information you can get from the RRD or RD or RD experiment. So to confirm this thing, we can do RRD experiment. So and this is a potential versus log of k. This is known, this equation is known as the Tafel equation. So although this is a little bit advanced, so I just mention all these things so that later you people can see, or when you will understand, read in the literature, then you can correlate with this topic. So I will share one article on this RD and RD. So it will be in the Google Drive, so you people can read one article on the Journal of Electrochemical Society. So that will be a good one good thing, good article. So basically this is the Tafel equation in eta, which is a over potential. So basically over potential, what is the over potential? So over potential is the potential or extra potential where the thermodynamically some reaction is going on, and from that reaction or that potential, how much potential is deviated, that potential is known as the over potential. So basically this Tafel slope or the Tafel fitting, you can get the get the over potential or slope, and from here also you can get the information about the alpha, which is the transfer coefficient. So basically from here you can get the information of transfer coefficient also, and these things, this is the JD, which is basically disk current, and this is the ring current. So basically whatever I have shown in the first previous slide, so basically you can fix the ring at the potential where the hydrogen peroxide can oxidize to O2. So this potential you can fix. So here the hydrogen peroxide is forming, as a result your current value is at the ring, your hydrogen peroxide concentration is increasing. So for this your current value is increasing, and after diffusion control region your probability is decreasing, so your current is depleting, and as the diffusion control process, so it is going on, and you can get from here the collection efficiency of your electrode. So how much collection efficiency, which is usually people write NC. So basically the ratio of the current density of the ring and current density of the disc. So from here you can get the what is called collection efficiency of your electrode. So these things you can get from this equation. So this is about the RRD and RDE experiments. So after that we will discuss about the bioelectrochemistry and biosensors. So basically one of the application of the electrochemistry. So basically the whatever in a living system some the electron transfer process is going on, for this some function is going on in your living system. So to study of the biological electron transfer process, or people usually keep one type of in vitro and in vivo study usually people do, that is one type of branch which is known as bioelectrochemistry. So basically the electrochemistry of the biomolecules, so that is the thing. And this electron transfer reaction play in this bioelectrochemistry all the biological system and the key role because all whatever process it is oxygen transfer or oxygen capturing or CO2 capturing from the living cell to transfer to the lungs, that's a process also depend on the electron transfer process. So basically different type of electron transfer process is going on in our living system. So to study or to understand that things usually people do by electrochemistry. So I'll discuss little bit on the enzyme or on important part of the protein living system. So basically in this biological bioelectrochemistry people usually use in biosensor application and biofuel cells and bioelectrosynthesis. So I'll discuss a little bit on this hemoglobin and myoglobin, which is one of the important protein molecule or enzyme in metalloenzyme in living system. So basically in the structure of hemoglobin or myoglobin, if you see there is a porphyrin ring, and this porphyrin ring on metal center is there. Basically if it is a heme, basically heme represent Fe2 plus as well as Fe3 plus so it is hemine. So if it is hemine, then Fe3 plus is there. So and this Fe3 plus and this is the myoglobin structure, where one hemine moiety is there, along with this the globular protein is there, and this is the hemoglobin. So hemoglobin Hb. So hemoglobin is the tetramer of the myoglobin. So people usually confine this protein molecule because this through the hemoglobin, which is a unsaturated part. So these things the electron transfer process from the electrode to this protein is difficult. So if you see your protein molecule in the bulk solution, and if this is your electrode surface, and from electrode surface to bulk solution, this electron transfer process is hindered because the electron transfer process is depend on the orientation of your protein molecule. So if you confine this protein molecule on the electrode surface and then this electroactive part of the hemoglobin is the hemine part. So here the iron center is there, it can go +2 +3 oxidation state to +2 oxidation state, and it can go to +2 to +3. So if you do the reduction or oxidation of hemoglobin in the bulk solution, then your redox peak reduction or redox process is an irreversible process. But if you confine this protein molecule in the electrode surface, then that is the redox process is quasi-reversible or reversible process. So if it is a quasi-reversible or it is a quasi-reversible process or irreversible process, there is some model which is known as you can say Leveron model. So from Leveron model, if you know, to the heterogeneous electron transfer rate constant and transfer coefficient, so these things you can measure from the Leveron model. As a protein molecule, the electron transfer process is a surface control process. So these things you can do. And as we all know that this myoglobin and hemoglobin, which is a oxygen carrier, so it can carry the oxygen from the lungs to your through the blood, and it can transfer the oxygen to your tissue cell. So this kind of application, so people usually get the idea that this kind of enzyme which can use in the energy storage as well as in the redox mediator as a redox mediator. So this kind of experiment people usually do in this energy storage, where you can see in this TOC that hemoglobin in the living system, where oxygen can capture and it can oxygen is transferring CO2, and from there the energy is releasing. So this idea it can convert to the metal air battery, which is one of the important energy storage nowadays. So because this metal air battery, the energy density is 10 times higher from the whatever conventional or lithium ion battery. So for this this air battery, one of the interesting what is called candidate for energy storage world. So here you can see here this hemoglobin which can transfer the electron, so it can act as a redox mediator in metal air battery. So this energy storage application also, this hemoglobin and myoglobin, this type of molecule can be used. So this thing is there, and now we will discuss what is the application of this biosensor. So one of the biosensor is phenolic biosensor, where you can see that in wastewater one of the important part of the environmental science or environmental control, that phenol can mix with the water, and it can toxic the water or drinking water. So to know the what is called percentage, or to know the particular the phenolic phenol is there or not, that time this phenolic biosensor is applicable. And this phenolic biosensor also applicable in the medicine and biotechnology. So this medicine and biotechnology also in blood or to determine the catecholamines, people use this phenolic biosensor, and to find the food quantity control, quality control also. Food quality control people also use this analytical chemistry to know how much phenol is there in the olive oil or red wine or milk product. Because so these things people usually use this phenolic biosensor. So one of the other overall application of the analytical science is that whatever we have discussed from the earlier class, that different type of compositional information and the structural information of material chemistry as well as biological chemistry or biochemistry, and to determine the quantity and quality of the analyte. You can use this analytical chemistry or analytical expert to know the better understanding of the to understand about the composition of your analyte. And other thing, it is also applicable in the forensic science and bioanalysis and clinical analysis, whatever in diagnosis of blood sample or material analysis. So people in industry also people use this analytical chemistry in the application of analytical part. And also statistics and solve the problem of analytical chemistry analytical purpose. Different type of instrument people usually use so to know or understand or analysis the data of whatever you get from the analyte. Different type of software or statistical method people use for know the analyte. So these are the application part of electrocardiography or EEG or ECG. Different type of application people use this part. So basically one of the major part of analytical chemistry that it is not as I am we are discussing that chemistry, but it is not limited on the chemistry only. So it is applicable in different field from materials engineering or materials chemistry to physics as well as biological system. So all these things we know. After that here I am completing the discussion part of this part or last class. So now we will discuss some of the question of previous year. So this is the first question. So here I will wait for, I think this is all are easy questions, so I will wait for 20 seconds or maybe. So you people can write in the chat box so that I can correlate. Okay. So the first question is what is determined by the Clark oxygen sensor. So dissolve oxygen in the blood, dissolve CO2 in the blood, dissolve CO2 in other body fluids, total amount of compounds present in human body. So if people can answer or write in the chat box. So as I have discussed in the first one, so basically what is there that it can dissolve measure the dissolve oxygen in the in your okay. So my laptop charger is not here, so I'll discuss a little bit fast. So next question is what is the correct statement for RD rotating these electrodes. So it is a hydrodynamic working electrode, as I have discussed. Also this is a hydrodynamic electro process, and this electrode can rotate during the experiment. So it is, there is some motor, so it can rotate the electron. It is used perform steady state studies of the redox reaction. So the answer is all of these. Okay. So next question, glucose plus oxygen hydrogen peroxide and gluconic acid is forming. So what enzyme is used for this reaction? So there is four options. One is maltase, glucose oxidase, trypsin and catalase. So basically if we discuss one by one, so maltase, what is the function of maltase? This is hydrolysis maltose to sugar molecule. Glucooxidase, which can transfer glucose with oxygen to hydrogen peroxide and gluconic acid. And trypsin, what is the function of trypsin? This is the trypsin enzyme which can what is called break protein molecule to amino acid. And this catalase, what is the function of catalase? Basically catalase, it can transfer H2O2 hydrogen peroxide to O2 and water molecule. Okay, so this is the function of reduce this is the function, so answer is glucose oxidase. Okay, so question number 4 is what are the membrane of the electrochemical glucose sensor more complex? Okay, so I the charge is complete. Okay, so the next question is why are the membrane of electrochemical glucose sensor more complex? There are four options. Outer membranes are permeable to glucose, impermeable to protein and other constituent of bloods, and consist of three layers and prevent enzyme from leaching out. And D option is all of these. So this question, I think if you watch the video then you can easily answer. So whatever we have learned from the class is that this is the glucose sensor electrode, where there is consisting of three layers. So the outer layer, which is formed of polycarbonate film. So this one is a form of polycarbonate film. So polycarbonate film, basically this membrane, outer membrane, is permeable for glucose, and this is impermeable for proteins and impermeable for proteins and other constituent of blood. And so that what is called the middle one, which is permeable for what is called hydrogen peroxide as well, and it traps what is called enzyme. So this one is traps enzyme, and the inner part, which is permeable for what is called hydrogen peroxide. So the electrochemical glucose sensor, whatever is there, its outer part is permeable for glucose and it is impermeable for proteins and other constituent. And as I mentioned, that it is consisting of three layers three layers, and this part is trapping the enzyme. So the answer is D, all of this. Okay, so go to next question. The next question is which of the following option is correct for phosphorylation? So the first option is ATP converts into ADP plus pi. ADP converts into ATP. So ATP means adenosine triphosphate and ADP is adenosine diphosphate. Glucose is produced and AMP is produced. So what is the answer? So basically adenosine triphosphate and this is adenosine diphosphate. So phosphorylation, basically the attachment of phosphate group to adenosine diphosphate. So the adenosine diphosphate to conversion of adenosine triphosphate. So the answer is B. Basically the attachment of adenosine diphosphate what is called phosphate group to the adenosine diphosphate is
energy, the energy for attachment of phosphate group to adenosine diphosphate is much higher than compared to first and second phosphate group attachment with the adenosine. So whenever the energy is available, that portion the conversion or phosphorylation reaction is happen. So the answer is B. So adenosine diphosphate is converted into adenosine triphosphate, which is this process is known as the phosphorylation. Okay. So the next question is, outer layer of electrochemical glucose sensor is thin film of KCl solution, cellulose acetate membrane, polycarbonate film, and glucose oxidase enzyme. So I think this question just now we have discussed. So basically whatever we have discussed, that this one is polycarbonate membrane, which is permeable for glucose, and it impermeable for protein and other constituent of blood. So the answer is C, the polycarbonate film. And whatever we have discussed, that the middle one, which is trap the enzyme, and the inner one, which is permeable for hydrogen peroxide, and it can go to the electrosurface. Okay, so the answer is C, that a polycarbonate film, the outer part. Okay, so the next question is, fill in the blank with suitable option given below. So hydrogen peroxide and what is called hydroxyl group, it is transforming oxygen and water molecule and plus 2 electron. So it is basically a oxidation process, as the 2 electron is removing. So the above reaction is take place at dash in the electrochemical glucose sensor. So middle layer cellulose acetate membrane, and inner layer and electrode surface. Okay, what is the answer? So middle layer, as I have discussed, so middle layer, which is the function, this is trap the enzyme. So this is trap the enzyme. And cellulose acetate membrane, basically cellulose acetate membrane is the inner layer. So inner layer, so basically inner layer, what is the function? So this one is the function is permeable for hydrogen peroxide. So it can so hydrogen peroxide can diffuse through this inner layer. So this inner layer, and it can reach to the electrode surface. At the electrode surface, this hydrogen peroxide, which can oxidize to water and water and oxygen in the presence of a hydroxyl group. So the answer is the electrode surface, and this reaction is happening at the electrode surface. So answer is D. So next question is, what type of bacteria desulfo vibriozigas is sulfate reducing bacteria, sulfate oxidizing bacteria, carbonate reducing bacteria, or nitrate reducing bacteria? So this one is the answer is this desulfo vibriozigas is a sulfate reducing bacteria. So the answer is A. So sulfate reducing bacteria, basically sulfate reducing bacteria is one kind of bacteria that can perform anaerobic respiration utilizing sulfate as terminal electron acceptor and reducing it to H2S. So basically sulfate group is reduced to H2S. Okay, so this is a sulfate reducing bacteria, as I have told. So the ninth question is, fill in the blank with suitable option given below. So O diphenol or catechol, whatever we know, that catechol in the presence of oxygen orthoquinone is formed plus water. So the above reactions take place in the presence of dash enzyme. So there is 4 options: monooxygenase, catecholase, kinase, and catalase. I think this question already we have discussed, that what is the function of different enzyme. So that we can know that So whatever we have discussed in the I think second or third question, that what is the function of monooxygenase. This is the function of monooxygenase is insertion of single oxygen atom in organic substrate. So what is the function of monooxygenase? So insertion of single oxygen atom into an organic substrate. Okay, so this is the function. And what is the function of catechol? So catechol is the enzyme that oxidizes catechol to or orthodiphenol to quinone. So the answer is this one. So basically this is catechol. Okay, so this is catechol or orthodiphenol, which can oxidize to orthoquinone in the presence of catecholase enzyme. What is the function of kinase catalyze? So this is kinase catalyze function, transfer of phosphate group from high energy phosphate donating molecule to specific substrate. So this is the function of kinase. And what is the function of catalase? Catalase basically it can decompose hydrogen peroxide to water and oxygen. So this one decompose hydrogen peroxide to water plus oxygen. So this is the function of catechol. So I think this one is already we have discussed in I think second or third question. There is similar type of another question was there. So the next question is fill in the blank with suitable option given below. So phenolic biosensor are used in wastewater treatment process, food like milk products, and medicine and biotechnology. And D option is all of these. So what is the answer? Okay, so whatever we have discussed for phenolic and biosensor, so basically it is used in wastewater treatment to detect the contaminants in the surface and groundwater, and in medical and biotechnology these biosensor are used to determine the catecholamines in blood, brain and cell culture, also used for detection of cell membrane and sensitive level detectors in enzyme immunoassays, and in food like red wine, milk to control the food quality. So the answer basically all of above. So these things we already discussed previous I think in the discussion part. So I think all this question we have discussed, and this is as I have mentioned in the starting of the class that this is the last class. So I hope the whatever we have discussed that will help to understand of this course, and whatever importance of this course in different field, that things we have discussed. So thank you all for joining, and for long time it stay with me. So that's all, and I hope you people will do good in this subject, and in future also you people can use this idea. So thank you all again once again, and I will stop here, and maybe and all the things whatever the study material and video, all this thing I will share in and upload in the YouTube, so people can access these things. Okay, so thank you, and okay, bye.